Okay, so we change gears here, and I thought what I would like to do is going back to the training camp. So the training camp is, I hope the trainees are at least getting the impression that this is about interactions, right? This is in essence trying to learn a lot about studying interactions, especially of GAGs binding to GAG binding proteins. And for that we designed the camp so as to utilize few very simple techniques to study these interactions, techniques that obviously we can do in our lab, as well as techniques that are pretty fundamental to GAG related work that you can essentially think of exploiting, and hopefully we can do some work together. And for that, the first training camp we had discussed, I believe we had discussed SPR. How many of you remember that? All right, very good. We did discuss SPR. Okay, so so I thought we are going to do SPR experiments here at the training camp, but I thought that's something that's already discussed. Therefore, I would not present on SPR. We had a protocol that we all did with regard to mass spectrometry, right? The training camps, remember? We did an experiment on mass and we had a little bit of discussion. So we have a little small mass UPLC-MS work that you all will be doing here. And I hope that you all recognize the pattern, the way in which the camp is designed, that there is a consistent pattern, and hopefully that will help you generate a picture that you will realize. I don't think really we talked a lot about fluorescence, so we thought it will be a good idea to talk about fluorescence. And obviously we never did talk about electrophoresis, especially capillary electrophoresis, that we are going to use. And we have both experiments planned, fluorescence and electrophoresis. Both are very simple experiments, so this is not really complicated. But I want you to think about utilizing several different techniques within these two broad techniques, methods that you could use, especially for GAGs, because GAGs are a different beast together, really complicated stuff. So so I thought I'll give you a background, and then hopefully you will appreciate the experiments that you're going to do in the afternoon. So a little background on fluorescence. So fluorescence is all about noticing energy released from the molecules, right, in the form of photons. So what typically happens is that molecules have electronic states. They have electronic states, some of the states that you might already know about. For example, there is a singlet state. For example, there is a triplet state, going back to really the basics. And then each of these states have vibrational levels. For example, these are vibrational levels. So this is the energy diagram of a molecule in general, and they have vibrational levels. And then within these vibrational levels there are rotational levels and things of that sort. So the molecules get distributed. So this is the S0 state, the ground state. This is the first excited state. So there are several. For example, you might have heard about pi star transition, right? Pi is a electronic state. Pi star, for example, is the excited state. So these are the states that we are really talking about. So molecules typically reside on the lowest vibrational level of the lowest electronic state, which is the ground state, majority of the molecules. And in essence, when we shine light, these molecules can undergo, if the light of certain intensity is not intensity, wavelength is present, then these molecules can undergo excitation. They absorb that energy. So for most part, from the ground state, you typically go to the higher state You can go either to the lowest vibrational level of the excited state or vibrational levels that are higher. And that's what we call as absorption, which is what you detect as the UV visible, for example, spectrum for most part. So this could be pi star, this could be n pi star, this could be things of that sort. This occurs really fast. This is essentially instantaneous. This is where, for example, the molecules just do not move. These are just absolutely stable. Nothing has happened. It's almost like the molecule is zapped and you have an excitation. So you do get absorption, tells you a lot about concentration, things of that sort. But for most part, that's not very useful. What happens when energy is absorbed? Obviously, it has to be released. So because it has to be released, there are several ways in which a molecule in the higher excited state can release energy. And one of them is simply thermal loss. There is vibration, the molecule keeps fluctuating, and all the excited energy is simply lost. It's dissipated, and it's gone. It's not really noticeable. That's typically true for most molecules. However, for some molecules, what happens is that it comes down from the higher excited state to the lower one through release of energy in the form of photons. Okay, so they actually release energy. The release of energy can typically arises from the lowest vibrational level of the higher excited state to any of the levels here. And that is called fluorescence. So this is what we typically utilize in numerous processes. These days, fluorescence is extremely utilized well. In addition to the general loss of thermal and the release of energy in terms of photons, if there is a state called triplet, which is possible for some molecules, you might actually transform a singlet state into a triplet state through vibrational changes and change in electronic structure. And then that could release energy. Obviously, that could also release energy because of thermal loss, but it can also release energy in the form of photons, and that is called phosphorescence. Both of these processes release light, and that can be detected, and that tells a lot about the system. That tells us a lot about the system. Allostery, for example, can be fairly easily detected using either of these processes. The reason is that fluorescence and phosphorescence are significantly delayed in comparison to absorption. They're 1,000,000 or 1,000,000,000-fold slower. So what's actually happening here is that the absorption, you are actually zapping the system, but in terms of fluorescence, the system takes some time. So by that time, the electronic states can actually change. The electrostatics can change. Structural changes can occur. And therefore, we get, depending on what the changes are, and because it's slow, that it allows those changes to happen, what photons come out can be detected, gives a lot about what the system is undergoing. Likewise, phosphorescence is even more delayed, and it can also give you a lot. But this is really a slow process. This is essentially like you hit somebody today and you get slapped back years later. So, okay. So not all molecules fluoresce. Okay, obviously, not all molecules fluoresce. Those molecules that have a reasonable transfer efficiency, reasonable rate of fluorescence do fluoresce. And there are these competing processes, for example, internal conversion, intersystem crossing, and then we can actually have some quenchers in our system. Because we are going to do the experiment, we always have to worry about these quenchers. One of the common quenchers is oxygen. Dissolved oxygen can also quench your fluorescence, so you really have to be careful. And obviously the concentration of the quencher. So this is the quantum yield. This is the quantum yield. You have to have reasonable quantum yield in order to detect what is actually happening to the system. Okay, so that's the principle. We have to detect photons released from the system. In addition to the thermal vibration and the temperature and the quenchers, there is also something in terms of the experiment that we always have to keep in mind, and that is the inner filter effect. The inner filter effect can also be a problem. So what is the inner filter effect? The inner filter effect is described here. So under normal conditions, we have to work with dilute solutions. Okay? We have to work with dilute solutions. We cannot really work with very concentrated solutions. The reason is that if the solution is dilute, whatever light that is incident, this is a dilute solution, this is a concentrated solution. So whatever light is incident, essentially the intensity of the light remains fairly constant across the path length. That means all the molecules that are present in the path length are getting exposed to essentially equal light. And that's important. Number of times there are certain chromophores, the molecule itself is such, or number of times because we do not know the concentration of the enzyme, concentration of the probe, accidentally we put, and we notice some change happening because of dilution, then we end up with wrong results. So because the concentration is higher, and as you keep increasing the concentration, only selected molecules get exposed to incident light. So what comes out is essentially nothing, and you get the wrong interpretation. So that's the inner filter effect. This is the inner filter. This is the high concentration inner filter. And then when you put your ligand, you dilute the solution, suddenly you start noticing that the intensity increases. And then you might end up with, gee, I have a beautiful effect. But then when you do it at different concentration, you have a different effect. And then when you do dilute, you have no effect. And 3 months down the drain. So you have to be careful about the inner filter effect. Okay, so here's some background on where should you look for fluorescence. So in essence, the excitation of the system gives us certain inference, gives us certain information. Most of the time the excitation is similar to the absorption spectra that we typically detect in a spectrophotometer. And it arises from ground state to various vibrational levels of the higher excited state. These excitations are typically detected as a spectra in a spectrophotometer. In addition, obviously, you can do the spectrophotometer thing also, and this should closely resemble the spectrophotometer. And that's the absorption spectrum, okay, where A is the least energy, this is higher energy, this is even higher, and things of that sort. Fluorescence arises from S1 to S0, wherein most of the fluorescence arises from the lowest vibrational level of the higher excited state. It can come back to 0 or 1 or 2 or 3 or 4. Where now this one is the highest energy and this is the least energy. So in essence, if you can, and by the time fluorescence occurs, there might actually be a small change in the levels of S1. So in addition to the regular case where all of this energy is lost, this part of the energy is lost when it comes down, that there might be a loss because of this change, that you end up with a fluorescence spectrum where these energies are always less than these energies. And this is logical, right? Whatever you put into the system is not necessary, you'll get 100% out. So always fluorescence spectrum is going to be on the lower energy side or the higher wavelength side of the excitation spectrum. Usually there is a difference between the A transition with a 0-0 transition on the excitation and the A prime transition, which is also the 0-0 transition on the emission, and that difference is called the Stokes shift. So usually there is a small difference. Not only that, there has to be some overlap. You can't really expect to measure this at one end of the spectrum and measure that at a completely different end of the spectrum with absolutely nothing in between. Typically doesn't happen. So this is how you actually measure the fluorescence. And most of the time, you either have an L-shaped geometry or T-shaped geometry. So this is an L-shaped geometry. This is the one that we have upstairs. And you have a light. Obviously, you have a focusing lens. Then you have a monochromator. This is the monochromator that allows you to select a particular wavelength, whatever wavelength that you want to use. And followed by that, you have a focusing lens which allows light to be incident at essentially the center of your sample. Okay. Now, this is in essence a single cuvette instrument. These days, obviously, we typically never use it unless we have very few samples to work with. We have high throughput screening assays in which this becomes a well. And the way in which you measure is also, the geometry is different. Light either coming from the top or from the bottom. And then the UV visible spectrophotometer would measure light that is actually coming out from here. So it measures the intensity difference between light incident and light coming out, and that is UV visible spectrophotometer. Whereas for fluorescence, light that comes out of the sample is measured on the right hand side, either here or on the left hand side, or both, so that you can follow multiple wavelengths. And what is coming out can also be recorded on a spectrophotometer, followed by detection using a photomultiplier tube. So this in essence is the rudimentary element of a spectrophlorometer. So you can actually change the selection of the wavelength either on the M1 side or monochromator excitation side or the emission side, and detect all different wavelengths, which gives you those broad profiles. Now, for most purposes, if you compare with NMR, fluorescence might actually sound like not good technique. NMR gives much sharper peaks, but fluorescence has its own advantages. Okay, so the advantages are that there are some natural probes that are inbuilt into the system. Tryptophan is the residue that is typically studied, although because it has significant quantum yield. Although tyrosine does fluoresce, phenylalanine does fluoresce, cysteine does fluoresce, but the quantum yields are not very good. You really have to label it dramatically. Cysteine, in addition, has a site that allows one to really label it with fluorophores, and that's another advantage. A number of times we put in cysteines in proteins, label it with something. DNA, for example, these adenosine and guanosines can also fluoresce. So there are these advantages that are already built in. Practically every protein would have a tryptophan. Just because the tryptophan is there doesn't mean the protein would fluoresce. You have to have some change. External probes are also fairly useful. So external probes can be small molecules. In addition, external probes can also be large molecules. External probes may be non-covalently utilized or may be covalently utilized. So either of those cases. And these are just a spattering, some things that I could quickly put together. But obviously this is just such a small number. In fact, there is a company that sells large number of probes and all over the entire wavelength spectrum. So for example, dancyl chloride or FITC or ethidium bromide, all of these are very useful. These molecules bind, the idea is these molecules bind the protein, and therefore the protein fluorescence will change. So these are not only useful for molecular purposes. Many of these probes are small and they are fairly hydrophobic. So you can actually use these to for proteins that are inside cells. So they are very, very useful. This gives you an idea about that you can actually utilize a large number of different probes such that the emission wavelength is in the visible region. So the idea is that whereas most of the previous probes, not all, but most of these probes have emission and excitation in the UV range, which is typically not easy to work with. There are so many different molecules that absorb in this region, and therefore it is not easy to detect small changes. So if it's completely shifted onto one particular side, then it typically helps. And therefore, we have many of these probes that are useful where the emission is in the visible side. Fluorescence is very, very useful because it is a very easy tool to find out the affinity, and this is the experiment that you are going to do. And you're going to use this equation, so I thought it will be a good idea to give you the background of this equation, just because I don't want you to be scared simply because the equation is big. The equation is big only because there are too many terms, but otherwise conceptually it's a very simple equation. So the idea is that the ligand, upon binding and formation of the complex, has to induce a change in fluorescence. That's the simplest thing, right? That's the idea. So without it, this won't really work. So there are a number of ligands where the protein has a tryptophan. They would bind, and yet they would not change. So keep that in mind. But the idea is that if a ligand changes the fluorescence of the protein, then that fluorescence change would be directly proportional to the concentration of the complex. And that direct proportionality would be maintained essentially at dilute conditions. Under dilute conditions, this should remain true. So what you would get is delta F is equal to, this is the proportionality constant. If we want to make it independent of the system, for example, the concentration of the protein, or the concentration or the size of the cuvette, or the aperture of the system, if you want to make it independent of all of these things, then you can actually derive an expression based on this alone, but then that becomes dependent on the system, whatever conditions that you are using. So it's a good idea to make it independent of the system so that we know what is the proportional change in fluorescence as compared to the starting concentration. And therefore, F0, just as delta F is directly proportional to the complex concentration, whatever the fluorescence was initially would be proportional to what is the starting concentration of the protein. And that proportionality constant is factored into this delta F max. So that's the fundamental equation. So out here we measure this experimentally. We know what is this. We can find out what is this. This is the experiment. We can find out what is this. What we don't know is what is this. That's what we don't know. Now this is dependent on the equilibrium, whatever the equilibrium is. So if we use the equilibrium as PL arising from P and L, concentration of PL is equal to P times L divided by KD. This is something that we saw in the morning also. And we substitute for P0 minus PL. The free concentration of the protein is total minus the complex. Free concentration of the ligand is a total minus the complex. And we get this. And if we solve for this, we get PL square is equal to PL times L0 P0 KD. P0 L0 is equal to 0. Now this is a very simple equation, right? This is an equation that we used to solve when we were in, I believe, 7th grade or 8th grade, right? This is a quadratic equation. So this is AX square plus BX plus C is equal to 0. That's what it is. And therefore we know that the two roots are minus B plus or minus B square minus AC, square root of that divided by 2A. A here is 1. Therefore there is nothing. And then B is this entire term. And then C is this term. So that's what this is. So PL is equal to, therefore, minus B and then B square minus 4AC. So if we simply substitute, that's the equation that we get. Right? So this is a simple derivation of the equation wherein what we don't know and why is it helpful in measuring the affinity. What we don't know is the KD. That's what we are interested in. And this we measure experimentally. This is what we decide what to put in. This can be measured experimentally. This can be measured experimentally. This is the same thing, and then L0 is what we know. So in essence, using this equation, this is the quadratic equation, we can find out what is KD and what is delta F max. Delta F max and KD are the two parameters that defines the system under the appropriate condition, right? So that's what it is. Sometimes delta F max can be high, sometimes it can be low, sometimes it can be positive, sometimes it can be negative. So you can have any of these combinations. That tells a lot about what it is. Delta F max gives you a lot about what is this in essence. This is in essence the difference between the protein in the unbound and the bound form. And structurally that can provide you some information about what is actually happening. Although if you really want to find out what is exactly happening, you need additional information. This by itself doesn't tell you everything. So this is very useful for yeah.
So this fluorescence delta F, is it intensity or wavelength maximum?
Delta F max is the fluorescence intensity at maximum. I'm going to show you. Okay. What is the change? So so that also tells me something that I forgot. So what we are doing here is the following. Okay, so what we are doing here is that in fluorescence we decide on which wavelength to utilize. For example, we want to use this particular wavelength in the affinity experiment. We decide on what wavelength to utilize, and then we decide on what wavelength to monitor the conditions. Okay? So we are deciding on a wavelength here and we are deciding on a wavelength here. So this is the excitation wavelength and this is the emission wavelength. So the excitation wavelength and the emission wavelength remains constant in that particular experiment. We are simply monitoring the changes in the fluorescence intensity. So Mausam, does that explain?
Yeah, I understand that.
Okay. Then?
Now the thing is, you said that intensity has to change after binding.
Yeah.
Now if the intensity changes, so there is some structural modification in the fluorophore, like.
If the intensity changes because of structural modification in the fluorophore.
Yeah, because it is all about fluorophore, right? The fluorescence has to come from fluorophore.
Okay,
So
go ahead.
The fluorophore part, like, is it like non-covalent interaction which leads to the decrease in intensity or increase in intensity?
Okay, so it can be non-covalent, it can be covalent, it can be either. Okay. So the simplest way to think about it is that if you have a protein, and let's say the probes that we had on the slide, those are aromatic probes. So the aromatic probes, let's say, bind in a site, right? Now these bind in a site. So in the free form, when there is water around, they have a different electrostatic environment. And then when they bind the protein, they have a different electrostatic environment, right? So the electrostatics changes. The structure, when I say structure, the oxygen, nitrogen, hydrogen, carbon, that doesn't have to undergo a change, such as oxidation or reduction. Nothing of this sort has to happen. They have to simply bind. The electrostatics changes. That change in electrostatics is what S1 transitions that we are trying to say, that they are different states now, and therefore it can be monitored, going from this process to this process. Now under certain conditions, you might actually have this complex already preformed, and then you introduce something like heparin. Okay? So if you already have a preformed complex and you introduce heparin, let's say binds to the protein on the other side, then it may be that this comes back out. Difference of these electrostatics of these two states changes, the intensity changes because the heparin binds. And or if this probe is bound here, because of heparin binding, something happens to the structure. Now the probe twists. That changes the quantum yield. It could be either. So we may not exactly know as to what is actually happening. Is it that less probe molecules are binding, or is it that there is a reorientation happening? We may not exactly know. But for affinity purposes, we simply have to have a signal. That signal has to arise because of interaction. That we have to assure. Without it, we cannot really go forward. Okay? Okay. So for GAGs, for heparin binding to antithrombin, this is a phenomenal piece of experiment because it allows us to measure affinities very easily. Here, for example, is the DEFGH that I talked about in the morning, and you can actually follow delta F by F0 as a function of the ligand concentration. And what you get is a profile of this sort. Okay, so in this particular condition, I don't really know exactly what this condition is, but in this condition, it actually reaches somewhere around 36% increase in affinity. So what we are measuring here is tryptophan fluorescence increase, okay, intensity of tryptophan fluorescence, and antithrombin has 4 tryptophans. Now, of course, we know as to what their contribution is and things of that sort. But for most purposes, this corresponds to increase in fluorescence arising from antithrombin, and this could arising from binding. So when the conditions change, you can actually see that the fluorescence changes. So sometimes the fluorescence decreases, as you can see here, and then if the conditions change, you can actually see an even lower change. The maximal change in fluorescence can be easily understood as that line, as that maximal line. So it's about 36% here, it's about 32% here, it's about 15, 18% here. So that's the delta F max. In addition, this is very useful because there is a visual identification of what the affinity is. Right? What is happening is saturation is happening at very low concentrations. Saturation is happening at very high concentrations. So the affinities are significantly different. Visual identification, approximately take 50% of that, find what the concentration is. That's the approximate affinity that you can measure. You don't even need a calculator. And you fit it to the expression, you get this line to give you the exact value of the affinity and the exact value of the Km. You have to remember that you have to reach reasonable saturations. Obviously, this is an extremely useful experiment because the delta F max is clearly defined. Okay, but number of times that's simply not the case, not possible because we don't really have enough GAG or ligand to really bind. So, how many data points do you really need in order to define this curve? You had to have 3 data points on this axis, approximately. You had to have 3 data points that define a curve. Then you had to have 3 data points to define this line, which is a linear profile. So in essence, you have to have a minimum of 10 data points. Higher the number, the better it is to reach the saturation level. 80% saturation is something that you should strive for, and that typically is believable experiment. Anything less is, you can always question it. This is also useful because not only can you determine the affinity, but you can also determine the stoichiometry of binding. So whereas for most purposes what we have seen is that the stoichiometry, assume that the stoichiometry is 1, the stoichiometry may not have to be 1. So what do I mean by stoichiometry here? The number of molecules that a ligand binds to the protein, the ratio of protein and ligand. So for example, if the protein is several subunits, you might actually have 2 subunits, each recognizing a ligand. So in that case, the concentration of the protein is 2 times more, the effective concentration, very similar to stoichiometric that we discussed in the morning. So in essence, the equation can be modified because the concentration of the protein now is 2 times. So therefore, it becomes n times p, and n times p. So what you have here is basically not only can we find out kd, but we can also find out n. Now those experiments have to be performed under appropriate conditions. So sorry. So for example, this particular case is non-stoichiometric binding. This is non-stoichiometric binding. This is stoichiometric binding. And the way that you figure out as to whether it is stoichiometric binding or non-stoichiometric binding is that there's a dramatic difference. So as you increase the concentration of the ligand, if you find that this much increase in the concentration is giving you this much change in fluorescence, then doubling the concentration is going to double the change in fluorescence. So all of this right up to fully formed complex, it in essence is completely linear, and right at that concentration, one of this 1 unit of ligand divided by 1 unit of the protein, if that is 1, you will basically saturate it completely and you will see no further change. That is stoichiometric titration. Okay? So you can use this equation for that profile and measure the N. Okay? So obviously this is useful because you can study as to how the different mutants can be affect the binding in terms of what is this? Okay, so this is antithrombin, and these are several mutants that we had prepared. I believe this is pentasaccharide. This is a 5 residue sequence, and under certain conditions it changes from 2 nanomolar to approximately 10 micromolar. Likewise, you can actually change the ligand concentration and measure the affinity so that we can determine the structure activity studies in really good detail. And this provides very useful information in terms of how our molecules bind. Now, the fluorescence change is also very useful, not just in terms of binding affinities, but it can also actually help identify multiple binding sites. A number of times you actually find that the delta F max is different for different conditions. And here is an example. So this is an unusual case, and thrombin figures in quite a bit, primarily because thrombin is really an unusual enzyme. So what was done was the thrombin was modified at the active site with a fluorescein label. So the thrombin itself is labeled. And then, although we keep thinking that heparin binds in exocyte 2, and that's it, and nothing further, here is an experiment where the fluorescence changes and then it starts to increase. And this increase can be, increases at a higher concentration, can really be fitted to a double quadratic function, not a single quadratic function, but a double quadratic function, where you have a concentration of thrombin and then a second molecule of heparin binds, a concentration of thrombin, heparin, ternary complex. And you can only imagine that if this binds exocyte 1, where is this going to bind? Most probably, this binds exocyte 2, where is this going to bind? Probably exocyte 1. So there are some recognition elements on both binding sites, and each of these have different delta F maxes. Okay, so fluorescence, this is something that you are not going to do, but in principle you can do fairly easily. You can actually use polarization and anisotropy, and this is especially useful when you don't really see a significant change in intrinsic tryptophan fluorescence as a function of ligand binding, but there is a possibility that you might actually see a change in polarization and anisotropy. So the principle is that light that is incident is typically normal light. It has all different vectors moving in all different directions. And you can filter out certain vectors using a polarizer, right? So you can actually make it plane polarized light in certain direction. And when plane polarized light is incident on our sample, then under normal conditions, if the sample is rotating fast, if it's tumbling really fast, then what happens is that there is mixing of this. So the plane is rapidly mixed, and you basically lose the directionality of the light that was incident on the sample. And that typically happens with small molecules. Small molecules move fast, they have a higher diffusion rate, and what comes out is depolarized light. So essentially everything is gone. So we are, however, for larger molecules that do not move very fast, that do not diffuse that do not rotate fast, that do not tumble fast, by the time the light comes out, it retains a lot of information of where it originated from. So because they don't move fast, the plane of polarization tends to stick, tends to carry forward, and we are able to detect what is this plane of polarization. There is some mixing that is involved, but to a significant extent that plane of polarization is retained, and we can actually measure different vectors, parallel vectors, these are all parallel, or perpendicular vectors, and based on the intensity of these we can find out what is polarization, and what is the change in polarization and change in anisotropy. And these are simple definitions. So if the light is incident in this particular direction, if this is the plane of polarization that is selected, and we measure light in the L-shaped geometry, this is the parallel intensity and this is the perpendicular intensity. Finding these two intensities, this is the total intensity, that gives us the polarization. As you can see that the maximum value is going to be approximately half. Anisotropy is slightly different. This is multiplied 2 times because there is a vector around z also, and that is taken into consideration, and so that is the total light, and it turns out that several calculations anisotropy is a better measure. So in the publications you would see anisotropy being used, but polarization and anisotropy can be interconverted. It's just a mathematical term. The max value is both of these terms should typically be positive, because the parallel is likely to be much more intense than the perpendicular. So it should typically be positive. So it goes from 0 to 2.5 or 0 to half maximum. So here's an example of polarization and anisotropy. This is one particular protein binding to yeast ARP23 in the presence of actin. And this follows exactly the same principle. So this is simply a response, just as the delta F by delta F0 is a response. And we can measure the increase in, and you can fit it to the complex quadratic binding equation. And likewise, you can do the reverse. So once you have reached the concentration, you can put in unlabeled protein and you would see a decrease. It turns out that this caught my attention simply because that the paper published recently tries to say that the actin presence or absence is making a difference in these two binding to each other. As far as the on experiment is concerned, which means the two molecules are coming close together, there's hardly any difference in the presence or absence of actin. But when it is formed, the way it is dissociating because of the presence of unlabeled, there's a significant difference in when the actin. So that's interesting in terms of mechanism. How is that happening? We don't really know. Go ahead.
Can you tell the size of the molecule based on the anisotropy?
Size of the molecule? Yes, polarization is related to diffusion rate. You can, diffusion rate is related to the size. So there are complex equations that you can do. I don't know whether you can do it in a static sense. This is a static experiment. But in a kinetic experiment, you should be able to figure out. D, yeah. Okay, another way in which fluorescence can be utilized is FRET. FRET actually stands for Foster Resonance Energy Transfer. It doesn't really stand for fluorescence resonance energy transfer. Most people think of it as fluorescence, but this is not really a fluorescence thing. Yes, there are fluorophores that are involved. So what is needed is now, instead of having one fluorophore, we actually need 2 fluorophores, and they have to be of certain type. So they have to be of certain type, and not only that, they have to be of certain distance, at a certain distance, in order to engineer the energy transfer that we are talking about. So for most part, the fluorophore 1 is able to absorb energy and is able to release energy. So this is the fluorescence of fluorophore 1. That is typically called a donor, and you can always only go from a higher to a lower. Therefore, this system has to be at a higher energy side. And then you have a fluorophore which has an excitation spectrum and which has an emission spectrum. When the excitation spectrum of the second fluorophore, which is called an acceptor, overlaps with the emission spectrum of fluorophore 1, which is called the donor, then when you excite the system at the excitation side of fluorophore 1, then what you detect is here. You don't really detect intermediate state. So what is happening is that the system is shuttling energy. You put in this energy, but what you get out is somewhere here. So you are actually shuttling the energy from this system to that system, and that tells a lot. The way in which these spectra overlap, the orientation, the dielectric constant, lots of these kinds of things play a role. In fact, distances up to approximately 80 angstroms can be really measured, and 80 angstroms is a fairly long distance. Our traditional molecules, 100,000 Daltons, typically 50 angstroms, somewhere around that. So that really covers a big space. Not only that, this distance can actually be quantified. So you can actually quantify this distance based on what the two fluorophores are, the donor and the acceptor. You can actually measure the distance between them based on how efficient they are. So this is the optimal, the ideal R0 distance between the two species. And this has been exploited to a certain extent. So the theory was developed a long time ago. It was put to test in just about 1950s or so. And as far as our field is concerned, which is of serpins, a group utilized this in order to find out what is the structure. What is the structure? Not really the detailed atomic structure, but where the different units of the protein are, using this profile by cysteine modification of several fluorophores. We now have several different fluorophores. So these are the traditional fluorophores. Naphthyl and Dancel systems were traditional. Phtc and Tritc were traditional fluorophores. Psi3 and Psi5, these are the newer ones. Enhanced green fluorescence protein and Psi5. Cyan protein, it should be F. Cyan's fluorescence protein and yellow fluorescence protein. These are the newer systems that you can actually engineer, you can actually express. So you can not only do this on a molecular level, single cuvette level, but obviously this is heavily utilized in cellular assays these days. So localization of different proteins at different places in the cell. I thought I'll highlight simple things where GAGs play a role, and apparently there are quite a few publications in this aspect. This assay has been utilized for HNE. HNE is human neutrophil elastase, very important as far as inflammation is concerned. And heparan sulfate modifies HNE. Heparan sulfate, at least in our assays, heparan sulfate is able to inhibit HNE. Okay? And the reason why it caught my attention is because this is completely opposite to what we had detected in our lab. So heparan sulfate is very similar to heparin, and heparin inhibits HNE, and it's really, really powerful inhibitor of this inflammatory protein enzyme. So what they did was the FRET peptide, they prepared a peptide which was hydrolyzed by HNE using orthoaminobenzoic acid, which is the donor, and on the C terminus they had 2,4-dinitrophenyl ethylene diamine, which was the acceptor. So you excite at 320 and you observe emission at 420. And this is a 13 mer. So this is on the same chain. One is the donor, one is an acceptor, and they are too close. So what happens is the situation, to start with, the situation is quenched. You don't see much fluorescence. So cleavage, and when you separate, you start seeing an increase in fluorescence because the distance increases way beyond 80 angstroms. So heparin increases. It turns out that heparin increases both the kcat and the km of the substrate. This is an unusual observation as to how can kcat increase when km increases. Not only that, in our system, the reason why it caught my attention is because kcat actually decreases, at least in our system. So that's really interesting. Not only that, in this particular paper, FRET was used in stop flow to study the mechanism of inhibition. So both of these increasing, it's really unusual, and FRET turns out to be one novel way of doing it, an easy way of doing it. Here's a system that Project 3 people will probably appreciate. So they studied FRET analysis of CXCR4 with STF1 alpha, a chemokine in the presence of heparan sulfate. This is published some time ago, and I have not really followed up as to what happened, but I thought this also is completely opposite to what I thought was actually happening in the system. So they labeled the receptor with enhanced green fluorescence protein, and they labeled the chemokine at position 68 with Texas red. So these are the two labels. You have a donor and you have an acceptor. And what should happen is you are starting from wide apart, and as the molecules come close together, you should basically start seeing suppression of fluorescence, right? That's the quenching that is exactly opposite to what in the previous case. And then dissociation should basically release it. So what is happening here is that and this is time, okay, so this shows time. So in 120 seconds, if you look at the fluorescence intensity, then the intensity is quenched as one would expect because of association in normal buffer. But when you put in heparinase, this is in cells. So this is basically monitoring in cells. That's the main advantage of choosing these fluorophores, because you are moving it away from the normal stuff that is floating around. So heparinase should actually cleave heparan sulfate. So it should chop it down, and basically if heparan sulfate was contributing, then you would dramatically change how the kinetics of association. If heparan sulfate was contributing, then what is this? Oh, this is a different concentration. Okay, so these are two different concentrations. So if heparan sulfate was contributing, then you should see a dramatically different kinetics. It turns out, well, heparan sulfate doesn't do anything here. So it's not only in association with the same phenomena in dissociation. And then this, in essence, is the same thing where heparan sulfate, this is in buffer where heparan sulfate was added to the medium. So this is in cells. Heparan sulfate was added to the medium, and basically no change whether the heparan sulfate is present or not. So in essence, this is a result that is completely opposite to what I thought was actually happening. Heparan sulfate modulating the interaction and making it favorable and things of that sort. But yeah.
So these are in live cell studies. So couldn't some of these results that don't exactly go with what you think is going on be because you attached a giant fluorophore onto the molecule?
Yeah, that is always a complication if you attach green fluorescent protein, but those precautions are usually taken. You put in a linker, you separate the modules, things are different. The dynamics could change, yeah, theoretically, but no, you have more experience with this, so.
They had a fluorophore at 68, lysine 68.
16?
The lysine 68, right?
Lysine 68, yeah. Why there? Oh, I don't really know. It's just an easy place to put in a carboxylic acid. but 68 is essentially at the end, right? Yeah, it's at the C terminus, so it's practically at the end. So maybe they figured, wow, it's a good place. So this is an old paper, by the way. Okay, so how many minutes do I have? All right, not very many. Capillary electrophoresis. Now, this is a technique that is very useful for GAGs because it gives good information in terms of what GAGs are, and this is one place where most people typically go to. This in combination with, for example, UPLC MS monitoring of what composition is, forms really nice combination of techniques to figure out what GAGs are we dealing with, and I think that's really important. So so the basic principle is very similar to electrophoresis, right? In electrophoresis we have charged species, you put in electrodes and the migration of these charged species would change. Okay, as simple as that. It's just that now we have capillary. That's the only difference. All right. So capillary is really, really small. This is a microcapillary where the internal diameter could actually be somewhere in the region of 10 microns to, I would say, about 100 microns. So it's a really small diameter capillary, and because of this there are certain additional features that come into play, especially electro-osmotic flow. So that's an additional contribution that occurs only in capillary electrophoresis and doesn't happen in traditional or tube electrophoresis, if you want to compare capillary with a tube, which is much, much larger. By the way, it does happen in tubes also, it's just that it's not noticeable. So in capillary electrophoresis we have the wall, typically silicon wall, quartz, and under certain conditions, especially at most pHs, let's say pH about higher than 3, these walls, the silanol groups, are typically ionized, and because of that the sodium that is present in buffer binds and forms essentially an immovable layer on both walls. So there is a strong positively charged layer on both sides, and the buffer typically keeps flowing. So because of this, what happens is that overall the charge of the species becomes such that these things get dragged on to the negative electrode, and you generate a flow, and you generate a current, and that flow is called the electro-osmotic flow. This is essentially an osmotic flow generated by the application of the voltage arising from differential charges across these two layers, and essentially immovability of this particular layer. So that electro-osmotic flow gives a defined direction to the flow. So it's not just dependent on the migration of positive charges to positive ions to the negative electrode and negative ions to the positive electrode. It's not just that, which is the electric field migration. But what happens is the overall flow moves in one particular direction. So it carries everything together. So the positives would definitely be attracted towards the negative electrode, but even the uncharged moves towards one side, and even the negatively charged molecules would move towards the negative electrode. Okay, so that's the difference. And because of that, in essence, if we have a sample in which there are charged species, either positive or negative, and there are uncharged species, then you would see resolution, then you would see detection of all three species. Of course, for GAGs, this is really not useful because anyway GAGs are all negatively charged. But theoretically, you could detect all of these too in a single set of experiment. That's the advantage, and the advantage also is arising from the micro nature of the capillary. So, okay, is that clear? Yeah. So I thought I'll just tell you a little bit about the basic equation of capillary electrophoresis, and the reason why I point out is that although we tend to think that capillary electrophoresis and migration time that is everything, it's way more complicated, arising primarily because of electroosmotic flow. So in essence, the migration of an ion, or the speed of the migration of an ion, is dependent on the electrophoretic mobilities. This is how charged the species are. Greater the charge, greater the migration, as simple as that. As well as it's dependent on the field strength, right? So it's actually dependent on how much field. Higher the voltage, higher the ability to pull it towards you. And because of the presence of the electroosmotic flow, both of these form the constituent. So it's a sum of both of these. Electroosmotic flow is typically in one particular direction, especially under normal polarity conditions. In the other direction, it is 0. So the mobility is directly dependent on charge, and it encounters resistance from the medium. So that resistance is a function of the radius of the protein, some of the things that you might be interested in. So this is function of a size, and therefore the mobility of the medium is proportional to the charge to mass ratio. The charge to mass ratio, and that you can correlate with the diffusion constant and the zeta potential and the viscosity and the dielectric constant. So all of these factors play in a role and make this equation fairly complicated. Obviously temperature plays an effect too. And therefore combination of factors determines the migration of an ion. So much so that molecules with the same charge to mass ratio can actually migrate at different speeds. That's the advantage. So you may have a molecule with same mass and same charge, and yet it would migrate at a different speed. That's the advantage, which means isomers can be detected, which means you have a sulfate here and you have a sulfate here. Those can be easily detected. So as far as GAGs, this is the reason why this technique is very useful for GAGs. So this is a simple electrode, and here is the electrophoresis. Okay, so you basically have a traditional electrophoresis, you have a buffer, you have 2 electrodes, you apply a voltage. The voltage is typically very high. It's not 100 volts or 250 volts, it's several thousand volts. And then you have this capillary. This capillary is of this sort. It's a very, very thin capillary. On the internal side, this is polymer coated, but on the internal side, this has quartz. And then you detect. So you create a window out here, and you basically detect what is actually coming out. This can be of various lengths. So the advantage of this technique is that the resolution is pretty high. The number of theoretical plates is really high, and that arises primarily because of the different flow conditions. So under normal conditions, when we do chromatography or when we do electrophoresis, the flow is typically of this sort, and in this region there is intermixing that is going on. Molecules kind of diffuse, and therefore the baseline resolution is typically not obtained. Under electroosmotic condition, the flow is somewhat of this sort. It's really flat, and therefore there is very little mixing. And you can imagine why is that happening, because there is a push that is going on. That is also true. This is also true significantly for UPLC, which is why you get high resolutions with ultra high pressure. This I already said. And then the advantages, there are really small injection volumes. So you could be working with 10 nanoliter sample. That's extremely useful. It also creates problem because the sample has to be reasonably well defined. Then you can actually interpret it. So in 1 microliter, you can do several dozens of experiment, as simple as that. So if we have a cell extract, remove the GAGs from the cell surface, collect it in few microliters. GAGs are difficult because they don't really have a chromophore, but you can still monitor it to a significant extent. So it's very small volumes that you can actually monitor. Usually the analysis is 10 minutes. For GAGs, it can vary because of the complexity of the sample. You can usually utilize both UV and fluorescence these days. Fluorescence is very heavily utilized. Now we have numerous techniques that have been developed: zone, micellar, isoelectric focusing, gel affinity, things of that sort. I have few examples because we don't really have much time. And these days you can actually shoot in cells. So you can actually shoot in the capillary, you can shoot in cells. If you have a technique where you can label it, if you have a protein expressing green fluorescence protein, you can actually monitor it. So that's in the works, in the sense being developed. Okay, so let's skip it because I don't really want to discuss all of the different techniques. But here's the power as far as GAGs are concerned. Okay, and you're going to do the CE experiment on Friday, one of the CE experiments. So for GAGs, we are always worried about what is the composition, right? What is the composition of GAGs? What are the disaccharides that are actually made up? And you can use HPLC too. But CE is especially useful, obviously, because it requires very low samples. We can generate these disaccharides. In these disaccharides, the information about the uronic acid is lost. But for most purposes, what the authors did, recently published, what the authors did was they took 17 disaccharides. So this is heparan sulfate. This is chondroitin sulfate. Okay, there is a galactose here. And this is glucose, this is 1,3. So this is hyaluronan. This is hyaluronan. Okay. So basically they did all of these put together. And the reason why this is important is because when we have cells, the proteoglycans can be anything, can be chondroitin sulfate, can be dermatan sulfate, can be heparan sulfate, and we simply want to find out what is the composition. So we do a digest, isolate whatever that comes out, and then we can measure what these are. And you can see there's almost baseline resolution for essentially all of the species, which is pretty phenomenal. Now this is something that was done long time ago, and this is not just disaccharides, but this is tetrasaccharides as well as hexasaccharides of defined structure with fairly good resolution. And you can see that the complexity is huge. That some of the species are really not fully well resolved. But if we look at, for example, let's look at 13 and 1. So 1 is the fully sulfated disaccharide. It's the highest charge density disaccharide. And 13 is in essence also highest charge density disaccharide. So this structure and this structure is very similar to that structure. It's just the double. And yet there is a significant difference in the retention time. So that really tells you that same charge to mass ratio also comes out at, can come out at different places, which is a specific advantage of this technique. Here's something that is very useful if you want to find out what is the composition of what comes out from the cells. This is developed very recently, and what they did was they mixed several of these, and in one assay you can actually find out whether it is dermatan or heparin, hyaluronan, or oversulfated chondroitin sulfate, and things of that sort. So most probably we are moving in the direction of compositional profiling directly from cells using capillary electrophoresis. CE is also very useful for measuring affinities, and this is what you do in terms of affinity. So here's the equilibrium that you're trying to measure. And in this case, the antithrombin peak in CE was being monitored. The detector was set at antithrombin. So you're basically monitoring how much of antithrombin is present. As you keep increasing the GAG, the position of antithrombin changes. So this is still antithrombin, which means that the antithrombin, which is in equilibrium between the free form and the bound form, because of that there is a change in the position. So the migration is changing because of the presence of the GAG. And this difference in the migration time corresponds to the proportion of the complex that is formed. And then you can set up the equation. So this is the traditional Scatcherd equation. You utilize the Scatcherd equation, you get this plot. From this, the slope corresponds to the affinity. Negative slope corresponds to the affinity. So it's a fairly useful technique. And then you can take this even one more step further. For example, here is a mixture of GAGs. So these are low molecular weight heparins. So these are low molecular weight heparins, and in these low molecular weight heparins, for example, there is peak 1 and peak 2, and you can follow the interaction of these individual peaks with different ligands. And you can do similar analysis. It turns out that in this analysis, instead of getting a monophasic Scatcherd, you're getting a biphasic Scatcherd, which really means that the ligand binds to peak 1 with much higher affinity than it binds to peak 2. This is the higher affinity, this is the lower affinity. Things of that sort. So capillary electrophoresis turns out to be a fairly useful technique. In addition, there are it allows us to measure, for example, changes in conformation. So allosteric can also be detected, although you have to be careful in interpreting and not over-interpreting. So here, for example, is the heparin's effect on beta-2 microglobulin. So this is the protein that undergoes aggregation, or at least a form that is amyloidogenic. And you can see that the structure of the smaller aggregates changes, and you can easily follow it using capillary electrophoresis. So I think that's the end. Yeah. So in essence, that is a technique. You all are going to do one simple experiment on profile oligosaccharide chondroitin sulfate. Okay. Any questions? I guess you're tired. So, all right, let's stop here.